Hello, everyone. Welcome to the Global Primatology Virtual Conference hosted by Central Washington University. My name is Margot Hinge, and I will be moderating this session. So this session is with Dr. Erin Ehmke, and it will last until 12:30. So before we start, I do want to let you know that the session is now being recorded. So if you do not want to be recorded, then make sure to keep your camera turned off. And then to make sure that this is a pleasurable and fun experience for everyone involved, I am going to request that we all follow a few session rules. So only one person should speak at a time. Please be respectful. Everyone is encouraged to participate and ask questions. However, due to possible time constraints, we may have to limit each participant to only one question. Avoid speaking on behalf of others. Do not be insulting, or you may be expelled from the session. And then finally, please keep the chat clear of traffic and only use it to send questions. But with that said, if you want to ask questions verbally, you can do so probably towards the end of the talk to just make it easier and have the session run smoothly. And for that, you will need to use the raise hand feature so that we can unmute you ourselves. So I will put these rules in the chat in a moment, but now that we're all on the same page, I would like to introduce Dr. Ehmke, who is the Director of Research at the Duke Lemur Center in North Carolina. So Dr. Ehmke, take it away whenever you're ready.
Thank you so much, Margot, for the introduction, and thank you to CWU for organizing this conference and inviting me to be a part of it. It truly is amazing what we've learned we can do virtually, and this conference is a great example of that. So I wanted to today introduce you to the Duke Lemur Center, and I hope some of you have had a chance to come visit us in person or plan to do so in the future. But for now, I'm excited to connect virtually with so many of you in this field. My name is Erin Ehmke, and I am the Director of Research here at the Lemur Center. I'm going to give you a brief background on who we are and why we're here, and discuss resources that I hope you find helpful in planning your own research, hopefully bringing you here to the Lemur Center. Now, the DLC is a world-renowned research center, a living laboratory of sorts, the only place in the world where such a breadth of research can be conducted on our earliest primate relatives, the lemurs. The DLC makes possible more than 100 scientific studies a year from all levels of researcher from affiliations around the world. And in our history, we have contributed to more than 1,300 scientific publications. Now, I'm going to first begin by, there we go. I'm going to first begin by sharing how our Lemur Research Center came to be here in Durham, North Carolina. It all began with a chance meeting between Dr. Peter Klopfer, who was a Duke professor. He studied parental behavior in mammals, mostly in farm animals. And he happened to meet John Buettner-Janich, who was a Yale professor who studied primate genetics. And they just happened to meet through the civil rights movement. It's a rather fascinating story. And through that chance encounter, they realized a really strong possible collaboration. John Buettner-Janich was looking to rehome his colony of lemurs, and Peter Klopfer realized that lemurs would be an ideal study system for his study of parental behavior. So he offered to help encourage Duke to provide a home for the lemurs here in the Duke Forest, and we've been here ever since. Now, when we started in 1966, our name was the Duke University Primate Center. In 2006, our name changed to the Duke Lemur Center, and that was done by our former director, Dr. Ann Yoder. She thought the new name better represented our focus. And in my opinion, it also makes it realize that we're not a biomedical facility, which most primate centers, or many of them are. The work that we do is very different. And so she thought that the new name better contributed to what we truly do here. Now that I've given a brief story on how the Duke Lemur Center came to be, I'm going to give you a brief description of how lemurs came to be and how they differ from the haplorhine world. I realize most of you listening have a lot of education in primate evolution and diversity, but I felt compelled to throw this in. So the primate story begins about 65 million years ago when an asteroid hit the Earth and created a cascade of events that decimated a large majority of species on Earth, at least 75%. Now, the silver lining to this mass extinction is that it created new opportunities for the species that did survive, including the small rodent-like mammals of the day. With most predators and competitors now extinct, these creatures were able to fill new niches, diversifying and thriving into the age of mammals. And this is where we see the lemurs arise as the earliest extant group of primates. And because there are about 30 million years separating them from the haplorhines, the monkeys and apes, we see a number of differences that are attributed to their more ancestral state. So as you see here, or I don't know if you can see it with the camera blocking it, but I have representing the human lineage David Herring, who is our former photographer and registrar. And I had him in this talk just to remind myself to thank him because all of the lemur photos in this talk are his that he took here at the Lemur Center. All right, so there are several differences, both anatomical and behavioral, between the stepsirines and the haplorhines. A few that I'm going to focus on just quickly. The elongated snout, the rhinarium, the wet nose, and the postorbital bar instead of full bony closure protecting the eye, all speak to the fact that lemurs are more primitive and more like other non-primate mammals, and that their primary sense is the sense of smell rather than eyesight. Lemurs are also distinct from most other primates in that most species do have a tooth comb, which is where the incisors and the canines of the mandible are fused together and jut forward. They can kind of use it as a shovel when grooming themselves and their group mates. And most species also have a grooming claw on that second digit of their hand. Now, behaviorally speaking, whereas in the monkey world, there's only one nocturnal species, there's no nocturnal apes. In the lemur world, there's a huge diversity. There's a good number of nocturnal species, diurnal species, as well as many cathemeral species. And the one thing that's really, really unique and cool about lemurs is that almost all species are very strongly female dominant. And the females do rule with an iron fist. They are not shy about showing it. And the boys certainly know their place. Now, this map shows in black areas where primates currently exist around the world. Now, when speaking about lemurs, they're endemic to only one place on Earth, and that is the island of Madagascar. The current species count is at about 110 species of lemurs across the island. And unfortunately, Madagascar has one of the highest rates of deforestation in the world. So we're at risk of losing all 110 species. So that's why the work that we do here at the DLC is so critical. Now here at the DLC, we currently have about 250 animals representing 15 species, some shown here. And these include a good mix of nocturnal, cathemeral, and diurnal species. All animals in our colony participate in our research program in some way, mostly through observations. All right, now the Duke Lemur Center has 3 missions, all of which are equally important: conservation, education, and research. I might be a bit biased because I'm the director of research, but I feel like it's research that's really the foundation to our other missions as well. It's the research that we learn how to better educate the public on how amazing these animals are and why they need to become engaged in conservation. And it's through research that we learn what variables are most important to focus our conservation efforts and our conservation funding on, what variables most impact their health, their social structure and dynamics, and the reproductive capabilities in the wild. All right, so why do we have a research center focused solely on lemurs? Why are lemurs so important? Well, as I've already said, they are so different. And because they are at the base of our primate family tree, by studying lemurs, we learn more about primate evolution and the changes that have happened since they evolved, and the biological processes that have happened and occurred through the primate order and during evolution. But also, it's equally important to learn about lemurs themselves, as I said, so that we can better conserve them in the wild and better care for them in captivity. And actually, we've learned a lot about improving husbandry practices through our research program here at the DLC. Okay, so who studies here? Well, anyone and everyone. We welcome all levels of researchers, from faculty to professionals, postdocs, graduate students, and undergraduate students. And we even welcome high school students as well, who are really engaged and interested. In my time here, I've been here about 10 years. I've been really focused on encouraging more and more undergrads to become involved in the research process. If they have any interest in it and want to know if this is something they want to do for the rest of their careers, I really encourage them to come out here and get their feet wet. And so for that mission, I created the Research Intern program. So in a normal year, in a non-COVID year, we would have our summer interns, and I also welcome interns through other semesters as well, where they learn the research process, and I mentor them through independent studies focused on our free-ranging animals. So they can really gain more experience and see if this is something they want to pursue further in graduate school or through field studies. Now, one initiative I'm really proud of was in 2019, I hired our first staff scientists. For our entire history, for more than 50 years, we've accommodated external research, and we will continue to always do this. But I really wanted to bring on our own scientists where we can start asking our own questions and leading the field in various ways. So in 2019, I hired Dr. Lydia Green, who heads up our microbiome and nutrition science program. And she's really studying the evolution of felivory. And I also hired Dr. Marina Blanco, who studies hibernation in the dwarf lemur. And I'm going to tell you more about the hibernation program at the end of this talk. I'm hoping as this program becomes more and more successful that I'll be able to continue hiring more scientists in more fields of study. Now, to talk about resources that we have available for researchers, we do have on-site housing available, again, in a normal non-COVID year, where researchers are able to rent a room in our on-site cabin. So they have ease of access to our center, and it's relatively cheap housing compared to other housing options. So if you are coming from out of area, please know that we do usually have that available. So inquire with me about that. And the other really exciting thing I wanted to mention is that we do have funding opportunities available as well. So you can check out our website for those descriptions. For graduate students specifically, as well as professionals and postdocs and faculty, we have the DLC Directors Fund, which will fund up to $10,000 per project. So check out our website for those funding opportunities that can be used to apply for research funding here. I did want to throw in here, talking about who are our researchers. We have had 2 CWU grad students come here in previous years to do studies, and they are both amazing and did really cool projects. So Carly Batiste came in 2017 and did a study looking at puzzle boxes, and it was a really fun project that she was here for several months over the summer. And then in 2019, Sean Mayo came, and he was looking at hand preference and the ii, really unique primate. And so his was a really fun study. So I hope we get more and more CWU students coming here to the DLC, as well as students from all universities from around the world. Okay, let's finally talk research. So I know some people get a little comfortable when they hear the words primate research, but I'm here to set your minds at ease. We are a research center that's open to the public. We wave that research flag proudly and with full transparency. And we can do that easily because all of the work we do is non-invasively. These are endangered species. Their care, their welfare is our utmost priority. We would never allow research that causes harm. Now, much of what we do is purely observational, where researchers come from around the world to follow our lemurs through the forested enclosures, watch the lemurs being lemurs and trying to figure out why they do what they do. Now, honestly, it might be more accurate to say that the research we allow is minimally invasive, because we will allow blood draws. For example, our vet staff will do blood draws for approved protocols that have merit to do so. And we also will allow projects that ask the lemur to do something. So I'm going to get back to this picture on the right a little bit later, but I wanted to throw this out there because if taken out of context, this picture could look kind of scary, like we're forcing some really uncomfortable things on the lemurs. But when you hear more about the research that's being done, you'll learn that it's actually not scary at all. It was a purely voluntary participation on the part of the lemur, and it actually really benefited him as an individual. And I'll explain that more later on. All right, we love it when research can be enriching for the animals, something that's fun and stimulating, and gives them something challenging and rewarding to look forward to. And we are able to foster this relationship through the use of behavioral training. Now, when asking a lemur to perform a specific task, we use training to teach them the goal. Then it becomes a game where they learn to voluntarily participate in exchange for a yummy treat. And when the lemurs are relaxed and not stressed, the researchers are able to collect high quality data in a relatively short amount of time. This picture shown here shows Meg Dye, our curator of behavior and of behavioral training, and she's demonstrating voluntary kennel training. So both of these lemurs voluntarily kenneled themselves, would allow the doors to be shut, and we could bring them in for a vet exam. That's a lot less stressful on the animal than having to chase them with a net and force them into a kennel. So all animals in our colony are trained for both husbandry and research purposes. So we started incorporating training into our research program about 15 years ago. And I'm going to show you a quick video of this origin. We had a researcher who wanted to study how shafak locomote and how their bodies adapt to the form of bipedal skipping they do when they come to the forest floor. To measure the impact on their lower limb joints, she placed a forest plate in the middle of the forest and waited for a shafak to skip across it. Well, of course, that's never going to happen. So we brought in a trainer, Meg, who you just saw. At that time, she was a consultant. Now she's on full time. And she trained our staff to train the lemurs to follow a target. Then we could lead the lemurs across the forest plate in exchange for a reward. The shafak's favorite reward is a peanut. Then it becomes a game to the shafak, and we get a lot of data very quickly. So I'm going to show you this video. So you'll see that green ball. That's the target. And here's the shafak in slow motion. Boom, boom. 2 really good footfalls directly on the force plate. Now the shafak will get a peanut, and now we can go back the other direction. And now within 10 minutes, 15 minutes, we can get a high quality data set, whereas before she would have waited months and months and months and not gotten a single data set or data point. And that lemur had a lot of fun. Again, this is voluntary. If the lemur is stressed or if they just aren't into it that day, they just get to go back home, or we stop trials. Nothing is ever forced on the animal. A few other examples of research that we've utilized training for. One area of study looks at sensory behavior. The top picture is showing a really interesting study looking at color vision capabilities of eyes and what levels of moonlight are needed to distinguish those various shades. This was a complicated project to train for. We had to train which color card was the correct card, and then they were given a choice of 2 cards to touch the correct color card that we wanted them to distinguish. And then we varied the levels of light to mimic moonlight to see when they could no longer distinguish the correct card. Now the picture below shows a different sensory project. This one does not involve training, but I wanted to show it because it's one of my most favorite studies we've done here, and the eyes had a blast doing it. So this was actually an undergraduate project, and it was looking at the behavioral preference for alcohol. Now, certain species have a mutation of the ADH4 gene, and those species that have that mutation are better able to metabolize alcohol to extract the calories from it. And those species that have that mutation have a behavioral preference for alcohol, like humans, chimpanzees. Eyes have this mutation, but they've never been studied in terms of do they have a behavioral preference for alcohol. So what we did is the student designed a study where we had a table set up with 8 different cups. There was a little hole cut in the top of each cup so that they could just sniff or put their tapping finger in just to extract a drop. And half of the cups had a control, just sugar water, and half mimicked fermented nectar. One of the favorite fruits of eyes is the traveler's palm. And so if they're seeking out fermented fruits, like many wild primates do, are they trying to get in more calories, more bang for the buck, right? Extract more calories, especially you can see how this would be really adaptive in food scarce times or food or resource limited times, where they're trying to acquire more calories at each food source. So the other 4 cups on the table had varying quantities of an alcohol-like substance. Again, there was very little quantity and a very little concentration in the cups. Of course, we did not want to affect their health or their behavior. We were just looking for their preference and if they could distinguish different concentrations of alcohol. So you could see the eyes would come down onto the table before we even finished setting up. They love this study. So what they would do is fascinating. They would systematically go around to each of the 8 cups and sniff or extract a drop of their finger. Once they had smelled all 8 cups, without second thought, they would go back to the highest or second highest concentration and focus on extracting more from those cups. So they clearly could detect a difference in the concentrations, and they showed a preference for that higher quantity. So it was just a really fun study that the IIs had a lot of fun participating in. All right, some other work that we've done that involves training. Energetics. I'm going to get back to this picture that I showed earlier. So energetics, this was a study looking at how various forms of locomotion affect their energetics. How energetically expensive is it, for example, to be a quadruped versus a bipedal leaper? And there was a lot of training involved for this study. So we first had to train the lemurs to be comfortable in a box, right? And this box was attached to very specific hoses that would measure the ratio of carbon dioxide to oxygen. Now, if the lemur was at all stressed and breathing heavy, it would have confounded the study. So we could only work with lemurs that were truly calm and comfortable in this environment. So a lot of training was involved. Once they were comfortable in the box, we trained them to walk on a treadmill. And then they also had to be trained to wear a vest. Now, of course, they couldn't put the vest on themselves, but they were trained to sit there and hold their arms out while we put their vest on them. So we didn't have to forcefully hold them. They would voluntarily put the vest on. And then they would voluntarily be still, while we shaved different parts on their limbs so we could put a marker, just a non-toxic paint that they would later groom off. And that marker allowed for three-dimensional cameras to recreate the structure of their limb as they moved. So a lot of training went into this to make sure the animals were comfortable. And why I wanted to show this was because this specific project really helped this specific animal, Terry's, a young male ringtail lemur. So at the time of this study, he had been recently kicked out of his family group, as happens with young adult ringtail lemurs. And he was having a difficult time acclimating to a new social situation. He became somewhat removed. He was starting to gain weight. He was kind of in a funk, at risk of sounding anthropomorphic. So we chose to try him for this study to see if it would kind of pop him out of that, if giving him a new challenge and something to focus on would really help his mental state. And it did. So we started working with him. He immediately engaged in research, was really immediately engaged in training, was really excited to participate. Through this research, he did lose a little weight, of course, because he's doing more exercise, and it seemed to pop him out of whatever funk he was in. We were able to better socialize him with Persephone, a female, and to this day, they're still together and very happy. So this was a great success story and how I believe research really helped this individual animal and his quality of life. Other studies where we've used training. This picture here, I have it showing cognition, and we have used touchscreen computers to study cognition before. This specific one is looking at sensory behavior as well, looking at color vision abilities, but it does also have a cognitive component. So it's really fun to train the lemurs to use computers and to do various cognitive challenges. Right now, we're even training our mouse lemurs, these little 60 to 80 gram tiny animals, to play on a computer. Now, they're too small to do a computer like that, so they use iPhones as their computer instead of larger screens. All right, to highlight some of the cognitive work, I wanted to show another video, just because I think it's both fun and fascinating. This was a project with a very simple design done by Brian Hare and his lab group, looking at the cognitive trait of inhibition, basically willpower. Are the lemurs able to control their impulses? So here's the setup. All right, there is, let me see if I can pause this without, yeah. So there is an opaque tube, one that they can't see through, and it has an opening on both sides. They can reach directly into it, either with their hands or with their face. And we would put a reward just in the middle of the tube with them watching. So they learn very quickly all they have to do to solve this challenge is to reach into the side of the tube and get their reward. We would do this training over and over and over until they learned what they had to do to achieve the reward. And then we would change out the opaque barrier for a clear one and see if when they could see the reward, would they still do what they learned was the key to solving the task by reaching around to the side, or now that they could see the reward, would they no longer be able to control their impulse to just reach directly for it? So here is a black and white rough lemur. This is the training phase. We did many of those, and once they learned, then we would switch it over to this test. All right, so she solved it, no errors made. Fantastic. Now here's the same setup with a cockerel shafak. Again, we did many of these trainings. And here's the test. Not the sharpest tool in the shed. Shavak are amazing animals, but they're not known for their cognition. Actually, what the group found was that it wasn't so much species differences, but it was more sex differences. Females tended to do better at this task than males, and that does make adaptive sense since females, their limiting resource is food. So females have to be really good at solving food-related challenges. All right. Now, other fields of study that we're able to accommodate in a more natural state without having to manipulate or ask the lemurs to do anything are behavior, social dynamics, biomechanics, how does the body move naturally, and communication. All right, now let me switch gears briefly to talk about another aspect of our research program, our biological sample collection. A collection that would be impossible to duplicate or replicate, as several species in our freezers are no longer in existence at the DLC or anywhere in captivity, at least in the United States. So the lemurs continue to contribute to science long after their natural lifespan. And through the use of these biosamples, we're able to accommodate projects that we would never allow on a living animal, things like viral studies and dissections and things of that nature. So how do we come about these samples? It's all opportunistic. Of course, we would never sacrifice an animal. So most samples are collected through routine veterinary exams. So whenever an animal comes in for a routine checkup, we will bank additional blood, serum, urine, feces, etc. And so through this, we're able to bank multiple samples at various time points over their lives. That's associated with very specific medical records as well as behavioral records. When an animal does die of natural causes or when they have to be euthanized due to quality of life concerns, we do save everything we can about that animal. So any organs that aren't needed for diagnostic, we will preserve. And the cadaver itself, we will bank in our freezers, and then we can provide those out to research as needed. And then, of course, I will take anything, anything that vets offer. So if they call me and say, hey, we had to extract a tooth or we had to amputate a finger or a tail tip, do you want it? I say, absolutely. If a husbandry technician calls me and says, hey, we found a sperm plug, would you like it? I say, absolutely. He wouldn't want a sperm plug. And so I have freezers full of these random, unusual things that you never know when they'll become important or interesting to researchers. And it's always really fun when I have a researcher contact me and say, this seems kind of random, but do you happen to have sperm plugs? And I say, absolutely, we have a bunch of them. So it's really fun. We have freezers full of stuff that if you think it could benefit you, just let me know and I can send you an inventory. All right, now I'm going to conclude my talk with an area of research that we're heavily prioritizing at the DLC right now, and that's hibernation in the fat-tailed dwarf lemur. It's amazing to think how this small species could have a huge impact on the understanding of the human condition. All right, so a little bit of a backstory here. The dwarf lemur is the only primate species that hibernates, and their hibernation is completely different from other hibernating mammals like bears. In the wild, they can hibernate for up to 6 months out of the year, and some populations actually hibernate underground, which is just really strange for a primate. They are primates like us, but they can stay completely inactive without losing functional muscle mass. Whereas if we stay inactive for just a few weeks, our bodies kind of start to melt, right? In addition, the dwarf lemur can go through rather significant annual fluctuations in body mass without developing heart disease or diabetes. So how do they do it? What variables prompt them to hibernate, and how do their bodies accommodate it? Through studies we've done here at the DLC, we've learned that extrinsic variables like food availability, temperature, may affect the depth and duration of torpor, but they don't prompt the animals to go into this behavior. So it's likely more of a genetic intrinsic trigger. So as primates ourselves, do we have the same genetic wiring for hibernation? We just need to figure out how to tap into it. If so, this could provide huge implications for the medical field. For example, putting coma patients into a torpor state to minimize cell death and muscle degradation. And if we're able to induce hibernation in astronauts, then we may be able to get to deep space. Now, the DLC houses the only captive colony of dwarf lemurs in the United States, possibly in the world. So we're in a unique position to untangle the intrinsic and extrinsic variables involved. The DLC has been studying hibernation for over a decade, and we're ready to take this research to the next level by doing more comparative work between experimental studies we can do here at the DLC with more natural patterns that we witness in their wild counterparts. So DLC scientist Dr. Marina Blanco is heading up this research program, and she has spent the last 15 years studying hibernation in the dwarf lemur, most of her work done in Madagascar. So to this end, we plan to build a field station in Madagascar to enable more structured comparative work between the DLC and Madagascar populations. The research will be constructed in Anjajavi, hopefully this year, pending the pandemic, so that we can study the genetics, physiology, and ecology of hibernation in wild dwarf lemurs. All right, so in conclusion, the DLC is committed to research and to serving as a model for how quality research can be done in a way that minimizes risk and promotes welfare of the animals, not only as a species, but as individuals as well. So if any of you have questions or would like to do your own research here at the center, please reach out to contact me and I'll be happy to get you set up to do so. And with that, I open it up to questions.
So if anyone has any questions, you can go ahead and either raise your hand to say it verbally, or you can always send it in the chat and I will read them off.
Hi, this is Natalie. Can you hear me?
I can.
Oh, I can hear myself, so yes. Thank you so much for the really great talk.
You're
I
welcome.
had a question about the research devices. So are the lemurs pretty neophobic with new items, devices, or things like that? Or are they pretty open?
It really
to
depends
interacting?
on the species as well as individual personality. So some species like the crown lemurs are more neophobic. It takes more training to acclimate them to a new device. Others are a lot more curious, like the eyes, even the dwarf lemurs. The rough lemurs are quite curious, as are the shafak. So it really just depends, and depending on what your project needs, our staff knows personalities of individual animals, and we know who might best work with the study. But we always, again, utilize training to acclimate them to any devices so that they are calm and comfortable.
Awesome. Okay, thank you.
You're welcome.
There is a question in the chat. So India Hicks is asking, so says, I would love to hear what your personal favorite species of lemur is and why.
That's a hard one. It usually is whichever species I'm most recently working with because I find them all fascinating. But in general, I'd have to say the eye, just because they are so smart and curious. It's really fun because you get to know them as individuals very, very quickly. They're a challenge to work with because they are so curious. They want to just be up on you and everything right away. And they have the potential to be very destructive. They can chew through anything immediately. So you have to really work and think outside the box on how to design a study where it'll keep your equipment, your devices safe, also the II safe, and how you can best ask your question. But they're fun to work with, so they're my favorite.
We should have a live question soon.
All right. Can you hear me?
Yes.
Yeah. All right. My question is also about my eyes. I run eye conservation. I wanted to ask, do you have access to an MRI scanner, and has there been any chance to run eyes through an MRI scanner to study their brain development?
Dr. Ehmke, you're muted.
Huh, wonder how I got muted. All right, so we have not done MRI scans of eyes. We do have access to an MRI scanner on Duke campus that they have outfitted for our small nocturnal species. So we have done MRI scans of mouse lemurs and dwarf lemurs, but we have not done any of the larger species. The main challenge with that is keeping an animal sedated long enough for the scan. So we need to make sure that it's safe for the animal. I would love to do one of eyes because their brain development is fascinating, but yes. Oh, you got muted, so I missed that last thing you said.
Oh, let me.
Very much so, especially because they do have somewhat color vision, especially for a nocturnal primate, and they have a very developed brain, almost on par with humans and chimpanzees.
Absolutely. That's what makes them so fun to work with.
Right? Exactly.
Yes.
Thank you.
Thank you for your question.
Okay, so we have more questions in the chat. Okay, so Jessica DeVille is asking, can you expand on the difference between torpor and hibernation?
Oh, good question. So hibernation is a longer version of torpor. So many animals will go into more of a daily torpor where their bodies might somewhat suspend or shut down in some ways for a few hours at a time, but they rouse again, within just a few hours or by the end of the day to eat and to sleep. Hibernation is a longer version of that, spanning over several days. So, for example, when the dwarf lemurs hibernate, they don't sleep, which is different from other mammalian hibernation. So the dwarf lemurs do have to rouse every few days in order to sleep, and then they can go back into their hibernation mode. So here at the DLC, we've been able to replicate almost normal hibernation and deep torpor patterns that you would see in the wild, where they can stay down for about 9 days before they need to fully rouse again.
Next question is from Elizabeth. Has there been any research about the effects of sunlight on nocturnal primate eyes?
Not that I know of, not that we've done here. So all of our nocturnal animals are on a reverse light cycle. So we somewhat trick them. So during the day here, we turn their lights out. They think it's dark to them. They think it's night. So they can come out, we can feed them, enrich them, do research with them. And then when we go home at night, their lights come on and they go to sleep. So we're very careful not to cross over those light cycles. We try everything we can not to expose a nocturnal animal to white light at an improper time because it can have dramatic and pretty immediate effect on their breeding cycle, as well as other welfare indicators. So we've never done that here.
Hi, Erin.
And then
I was wondering with the ongoing pandemic, if you've been able to open up to other research opportunities, or if it's just research that's been going on within the center.
Hey, Hannah. And Hannah was one of my former research interns, so she's fantastic. So we were closed down, March to July. We had no research going on during those months. We did reopen for research in July, but on a very limited basis. The lemurs are potentially susceptible to COVID through some studies not done here, but we did provide some biological samples that were worked on for some research groups that found that some of the lemurs are quite susceptible to COVID. So we've been very careful with bringing people on site, and we have very strict policies and regulations on who is allowed on site and when. So we have been able to reopen to research in some limited way. We have to maintain social distancing between humans, but also between ourselves and the lemurs as much as possible. And then, of course, additional policies on top of that. So the observational research, especially of our free-ranging lemurs, is the least affected because, of course, you can maintain distance very easily from the lemurs in that way. But some of the more hands-on work with the lemurs where we're asking them to do something, that has slowed considerably. We're hoping, that we're seeing hopefully we'll get back to normal somewhat soon. But right now we're still closed to research that can't abide by the COVID safety policies.
Next question in the chat. So from Megan Solis, are there any studies about the IIs that are ongoing right now at the DLC?
Let's see, right now we have an ongoing study that's been going on for several years where it's purely observational. They have cameras fitted into each of their enclosures. And this researcher is looking at maternal behavior of moms and their infants. Very little is known about infant IIs and their development and how moms raise them and teach them. So this is a study looking at that. We even have cameras outfitted in their nest boxes so we can see that. So that one has been going on for several years where, again, we don't have to do anything. It's just cameras fitted in and the researchers watching through that. We do have a study that's going to be starting up next week where we're just looking at food gape size and how they accommodate chewing different size food items, essentially. But right now, those are the only two projects we have working with IIs.
Thank you. Next question. Sorry, the chat window keeps moving. I have to go back up. So do you study the II at night, or do you manipulate their environment to study them during the day? And I guess you already kind of touched on
Yeah,
that.
we do manipulate their environment to study them during the day. We're hoping we can one day get to having free-ranging IIs out in our forest enclosures. We're working towards that goal. It involves a lot of logistical concerns that we have to make sure we can keep them safe in that environment. But if we get to that point, then it would involve a natural light cycle. So then we would be accommodating research at night. But again, we're not there yet. Fingers crossed.
So question from Andy, is there a database to access any of the research that is conducted at the DLC?
We do. I'm sorry, go ahead.
Just there's a second portion of this. So papers that have been written or anything like that.
Yes, we do. So there's two different spreadsheets, or one database and one spreadsheet that might be of interest to you. So on our website, we have a database where you can access various life history variables. So you can look through the history of our colony and look at weights, reproductive variables, things of that nature. And that's free to access. You just have to register. And you can download that database and get all kinds of more so medical and reproductive information about the lemurs through their lifespan. We also have a spreadsheet listed on our website. It probably needs to be updated, I'll be honest, so I can work on that, where we do list the history of projects, or maybe it's publications. It's more likely our history of publications. So from that, you can see the 1,300 plus publications that we've contributed to. So If you're interested in a specific field of research and you're not finding that information on our website, I'm happy to send you a listing of what we have done if you just reach out to me.
Great, and we'll make sure to list your, well, I guess your email is on your slides right now. So anyone interested, just make sure to write down Dr. Ehmke's email.
Yep, and if you go to our website, you can find me easily on the website as well.
Right. Next question. What are your thoughts about using red lights to study nocturnal animals, particularly for simians, to study their behavior in the wild or in captivity, like sanctuaries or wildlife rescue centers?
Red light is quite useful. We still try to minimize how long and how bright we make the red lights. So we've done studies in the past compared to other captive facilities as well. And red light seems to be the least invasive light to them during their night cycle. Blue light has found to have more impact on their behavior than red light. So we stick to red light and we only use it when needed and at very low levels. So we're still very aware that it could have some impact on them, but it doesn't seem to behaviorally affect them. They don't shy away from it. They don't cringe. But we try to use it very minimally.
Renee is asking, how do you form the social groups of lemurs, and how do you integrate new members to diminish possible intraspecific aggression?
It's hard, and that's really the work of our curator and our husbandry team, supported by our veterinary team, where if they want to integrate a new member into the group, they'll have the new individual next to the other individual or the other group in a adjacent cage for quite some time and just watch behavior between that barrier. And if all seems to be going well, there seems to be only positive interest and not negative interest, then they will start an introduction, of course, fully staffed with staff observing and ready to stop the introduction if need be. And whenever we do an introduction, our vet team is on standby in case there are any injuries. But our staff have gotten so good at reading behavior and knowing personalities that might mix that we rarely have injuries due to introductions anymore. Generally, we keep family groups together as much as possible. But of course, young adults are forced to disperse. And so when a parent is being aggressive to an offspring and trying to encourage that dispersal, we have to support that dispersal so that injury doesn't happen, because that's actually when we see more injuries is from parent to offspring. And sometimes it can be a slow, gradual increase in aggression over time. Sometimes it can be with a snap of fingers. Suddenly, one day they're great as a family. The next day, mom is really beaten up on daughter or dad's beaten up on son. And we have to do it very quickly. So we watch family dynamics very carefully. Try to follow when they're encouraging dispersal, and then we have very gradual and slow introductions based on behavior and how they're doing through caging.
Next question is from Jenna Skinner, who's asking, has Duke considered creating a primate program for students?
I'm not sure about that. Duke does have several classes that are supported by the Duke Lemur Center, and they're based out of here. So there's two classes in particular. One is field methods, where the class is based out of the Duke Lemur Center. They have class here, and they go out into the forest enclosures and learn various field techniques and do independent projects. There's other Duke classes that come out here for field trips or for data collection purposes. Some just come for tours. So Duke very much supports us, and we have a great relationship with many, many departments across campus, especially evolutionary anthropology and biology are our two biggest users on Duke campus. And again, several classes are designed to be done here at the Lemur Center.
Let me go through the list. Okay, So, question from Mayo, how are the eyes acclimating to the expanded outdoor enclosures?
Very well. So again, it's been a gradual process and it's been very individual. Some eyes have taken to the outdoor enclosures quite easily. Some were hesitant at first and slowly investigated more and more. So let me explain. So because we manipulate light cycle and have them on a reverse light cycle, historically the eyes have been kept inside, but we know they need more space than the indoor enclosures can provide, and we want to give them more visual stimulation, more olfactory stimulation of having access to outdoor enclosures like the rest of our, diurnal and cathemeral animals do. So as a pilot, we gave 4 eyes outdoor access. So it's not forest enclosure. It's still a caged environment, but it's connected to their indoor environment. So we didn't change their light cycle. We wanted to kind of give them the choice. So it's a little bit difficult to explain quickly, but we kind of melded the two, their manipulated light cycle with the natural one outside. We somewhat melded them and gave them the choice. And we still encourage them to come inside and sleep at night where it was light inside. We would turn on brighter lights outside at night to encourage them to come inside and sleep, and made it darker during the day for them to go outside. It's a bit hard to explain over quickly, but they've really enjoyed it, especially our younger ones. The older ones that have been in captivity and inside for most of their lives, they were more hesitant. But the young eyes that we gave access to these outdoor enclosures, they bounced right out and they were outside where mom was inside. I mean, it was pretty amazing. They were really excited. So I think we're going to keep continuing this program and giving more and more of our eyes outdoor access. I think that's a great, tremendous improvement for their welfare. And hopefully it gets us one step closer to being able to free range them.
Next question from Clayton Pilbrow. When will the stud book be updated next?
I don't know the answer to that one. I could find out for you though. Our curator heads up the stud book, or that's not actually true. It's not the curator. It's there's a stud book keeper for each stud book, and so the stud book is a list of all individuals and who their breeding recommendations are. And this is collaborated and maintained within the entire North American population. So here at the Duke Lemur Center, some of our staff are the stud book keepers for certain species. So we are the stud book keeper for Shafak, for mouse lemurs, and I think we are the co-stud book keeper for a few other species as well. So it's up to them on when they publish it, and I'm not sure when those numbers are. So if you reach out to me, I can put you in touch with the people that would best be able to answer that question.
Next question, do you partner with any AZA zoos for retiring any lemurs or acquiring new ones?
Absolutely. So most of the animals here at the DLC have been born here and live their lives here, but we do follow breeding recommendations. And if we need to transfer an animal out to another AZA accredited institution for breeding recommendations or bring an animal in, we will do that if at all possible, if we have the space and if it doesn't mess with our social dynamics by sending an animal out. We also have the flexibility to work with AZA institutions if, for example, we just need a partner animal. If another animal's mate has died and needs a companion animal, we're able to bring in companion animals as well. So absolutely, we work with many, many AZA institutions to further breeding recommendations and to optimize social dynamics and housing conditions.
Next question, how peaceful are lemurs compared to, for example, chimpanzees? Are there significant differences within various lemur species?
Absolutely, there's a lot of differences. So compared to chimpanzees, I would say, and this is not a joke, our most aggressive species is the mouse lemur. So they are the tiniest species we have, but they are super mighty and aggressive. Females really only have patience for males during the breeding season, and that's actually very limited. Usually after they're mating, they will attack the males and chew them up. So our mouse lemurs keep our vets very busy. They're always stitching up ears and fingers and tails. So we have to very carefully manage housing on mouse lemurs because they are quite aggressive with one another. Otherwise, the ringtail lemurs can be quite nasty. Those females are dominant and they are not shy about slashing up somebody else. So, I mean, I don't know how to compare them to chimps, but they can be quite nasty, and it really just depends on personality. A few of our, dominant females are pretty laid back, whereas others are not. Our blue-eyed black lemurs are also another pretty aggressive species with one another. But then you compare it to more laid-back species like the mongoose lemur, the red-bellied lemurs. They're a lot more chill, and they're more co-dominant. They don't have as much dominance hierarchy that's enforced, and so by that, they're a lot more laid back.
Tracy is asking, have you had any experience with ringtails diagnosed with lymphoma?
Yes. I'd have to talk to the vets to get specific types of cancers that are most prevalent per species, but we do see maladies that are common per species. And cancer is not fully uncommon just because we have gotten really good at keeping the lemurs alive very long. And so just like with humans, as we age and extend our lifespan, we become more prone, more vulnerable to age-related diseases. And it's the same thing with lemurs. So we do see various cancers. And again, certain species are more vulnerable to some cancers than others. We'll see kidney failure in some species like the bamboo lemurs. I'd have to get with the vets to get more specific than that. So if that's a question you have, let me know and I'll be happy to get that information for you.
So we have another question, and it might be the last one. Do you have any multi-center research project going on? Is there opportunity for staff at another facility with lemurs to participate in research?
Absolutely. So we have a lot of researchers that come here, and we are just one stop on their research design. They might look at us compared to a zoo, compared to a wild population. So we don't currently have any formal collaborations between the DLC and another institution, but we welcome all research. So any researcher from any other institution or center is welcome to come here and do more of their research.
And I guess I just remembered of a question that I had during the presentation, and I've gone through all the questions in the chat. So I guess I'll go ahead and ask my question. But you mentioned that there are plans to build a research station in northern Madagascar to study the dwarf lemurs.
Right.
I'm assuming there are other lemur species in that area. So are there any plans to also open up that research station for research with other species?
We would like to. One of the reasons we chose this location is because it does have, I believe, 9 lemur species, and many of them are quite comparable to the species we have here. And so it would be great for various levels of comparative work. And so we're building the research station for the initial focus of hibernation research, but we're going to design the station in a way that the enclosures can be modified to accommodate larger species as well. So we do plan to expand what we do at this research station, but the initial focus will be hibernation.
Thank you. And we do have a new question. How expensive will the new nocturnal research station be?
That's a good question. We don't really know yet. We're designing it to be very simple. We're basically designing an enclosure system very comparable to the enclosures we have here, primarily so that we can bring hibernating dwarf lemurs in for the season. So again, we won't be disturbing them when they start to hibernate. We'll bring them in, study their natural patterns, and then release them and track them once hibernation season is done and they're heading into breeding season. So it'll be a few enclosures designed very similar to what we have here. And then there'll be a human area where there'll just be a simple bedroom and meeting area. It's located within a private reserve, so any researchers staying there will be able to utilize the lodge for food and things of that nature. So because of COVID, we've had minimal communication on the logistics of building the facility. We're hoping it'll be no more than $50,000 because we did acquire a donation of $50,000 to build it. And so that's what we're trying to keep it to that budget.
Thank you. I think that's it for the questions. I'm not seeing any new ones, and no one is raising their hands.
Okay, if anyone thinks of any other questions, please feel free to reach out to me. I love questions.
Thank you so much, Dr. Ehmke. Thank you everyone for attending. This was a very productive Q&A session.
Thank you. It was a great opportunity, and I really enjoyed your questions.
Well, this concludes the session.
Thanks, everyone. Enjoy the rest of it.